Good day, ladies and gentlemen. Today we are going to look at topic 6, which is concerning red blood cells, and now we are going to dwell on the abnormalities of the morphology seen in red blood cells, and this is usually in a thin smear. My name is Dr. Penny Nansamba. I'm in COVAB, SBLS, Department of Biotechnical and Diagnostic Sciences, and I'm a virologist, both applied and molecular. Thank you. Our goals today would be 1, to describe the morphology of normal red blood cells that can be observed during the analysis of a thin blood smear. Number 2, to describe the abnormalities that we can see in red blood cells during the analysis of a thin blood smear. And lastly, to highlight general causes of the mentioned abnormalities in number 2. Our content is going to consist of the following: introduction, where we're going to look at the shape and the function of the red blood cells; evaluation of a thin blood smear, and here we shall be concentrating on the red blood cells. We shall look at the first abnormality, which is an abnormality of shape called poikilocytosis. The second abnormality, which is an abnormality in size, anisocytosis. And we shall also look at other abnormalities in staining, inclusions that appear, and abnormalities in distributions of patterns of these red blood cells. So, what's the function of the red blood cell? It is to transport oxygen to the tissues. And the red blood cells are so numerous that we count them in millions. They can be in millions per microliter, 10 to the power 6, or they can be in millions per liter, 10 to the power 12. And in the middle we have a cross-section of a red blood cell and also a micrograph of a red blood cell. It's biconcave and has no nucleus. Here we're showing you the biconcave nature of the red blood cell. We have special cases of amphibians and birds where the red blood cell is enucleated. That means it has a nucleus and it's not round, but rather it is long or elliptical. The evaluation of red blood cells in a thin smear. It's important that we evaluate the red blood cells during the analysis of a thin smear because it's going to tell us some abnormalities that will help us diagnose a variety of disorders. And this is going to support us in the interpretation of the red blood cell indices in the hemogram or the erythrogram. The morphology can be seen visually in a thin blood smear that has been stained with the Romanesque stains. And when evaluating a red blood cells, we look for the following: abnormalities of shape, abnormalities of size, abnormalities in color, whether the red blood cell has got inclusion bodies, it shouldn't have anything, not even a nucleus, whether there are abnormal patterns of distribution of these cells in a thin blood smear, they should be uniformly distributed, and also looking to see whether we've got infectious agents in the red blood cells. The first abnormality we're going to look at is poikilocytosis, or an abnormal shape. The micrograph on the left shows cells that are not round, they're sickle-celled. The micrograph on the right shows actually 3 different abnormal shapes. The cells called 1 are elliptical in shape. The cells called 2 have got actually a teardrop shape. And the cells called 3 are actually target cells. They look like a bull's eye where you're aiming for a target in the middle. Now here we're looking at red blood cells that have got different sizes. The micrograph on the left shows large red blood cells. We're going to call them macrocytes. The larger ones have got red arrows pointing at them, and the normal sized ones don't have any arrows. So these macrocytes may be as a result of anemia, hemorrhagic anemia. Now when you look at the micrograph on the left, the longer arrows show smaller sized cells compared to the rest. Those will be now called microcytes, and you can see them in iron deficiency anemia. Still on abnormalities of size, this micrograph actually shows very different sizes. Some are large, some are small, others are tiny. So all of these different sizes combined in a single blood smear would actually be labeled anisocytosis, different sized cells in the same smear. We're going to the next abnormality. This is an abnormality in staining. Here we're showing you red blood cells that have stained very faintly due to anemia, and we're going to call them hypochromatic, or we are seeing hypochromasia. Again, it might be due to iron deficiency anemia. So when we look at this slide, we can see some cells that are actually staining very, very light. We can see some cells that are staining very, very light compared to the darker ones. Here we're seeing macrocytes staining darker in the presence of normally stained cells. So this slide shows polychromasia. Now we're going to look at abnormal inclusions in the cells, the red blood cells. The first abnormality is basophilic stippling. The red blood cells are devoid of any nucleus and any ribosomal RNA. So if these are not extruded during genesis of the red blood cells and they remain, we can say that those cells have got basophilic stippling. It's shown by the blue arrows. The little black dots shown by the blue arrows, which are remnants of the ribosomes, is known as basophilic stippling. The second abnormality is a retained nuclei and is shown by the red arrows. Those are immature red blood cells that have been released into circulation that still have the nucleus. And we also have other abnormalities like the Howell-Jolly bodies and siderosis, which you'll meet very later. Now we're going to look at an abnormality in the pattern of formation of cells when you make your red blood cell, when you make your thin blood smear. Towards the left, we see that the cells are showing rouleau formation, and towards the right, we see that the cells have formed agglutination. This is a close-up of what rouleau formation shows in contrast to agglutination. The last abnormality we are going to look at is parasites. They can occur in the cells. They may be malaria parasites seen in the far upper left. They may be Babesia that's seen in the far right, or we may even see Hemoproteus seen in blood cells from a bird in the bottom left. So those are the sources of my information. And with that, we've come to the end of today's lecture. Hope to meet you in the practical session when you can actually identify these abnormalities. Thank you.